Okay, so this is part 3 for chapter 9, and it's the chromosomal basis of inheritance. So we're going to look how the chromosomes themselves can help pass on those genes. So we talked about loci, and chromosome theory of inheritance is explanatory of why genes occupy a specific location on those chromosomes. And then the chromosomes will undergo segregation, so they separate an independent assortment during meiosis. We talked a little bit about that. And Mendel's laws correlate with this chromosome separation. So it has to do with the separation of homologous chromosomes in anaphase 1, and the independent assortment depends on which orientation of those chromosomes in metaphase 1. So here's a couple of different examples. Look at what's going on to the left and look at what's going on to the right. If you notice on the left, the red is on the far left of the cell and blue on the right, and on the right it's mixed up. And then when they go through anaphase 1, they split, and then anaphase 2, they're going to split again. And so you get all sorts of different gametes depending on how these line up. Now imagine this, by 23 times, all the different possibilities going back and forth with 23 chromosomes. And so this was our dihybrid cross and the 9331 example. So when you put all those possibilities together and look at them, that's what you see. So Basie and Pundit looked at the plants that didn't show the 9331 ratio and said, what's going on? Why is this happening? And what they found was an example of a linked gene, in which all a linked gene means is that two loci or two alleles are so close on a chromosome that it's hard for them to be separated. And so they tend to be inherited together. So it's no longer that random assortment, right? It takes away from that randomness. So experiment, you got your purple flower and you got long pollen, and what you observed did not match what you would predict. And so what's going on there? And so you have your linked genes, and if you look at the location of the linked genes, it makes it more difficult to get the standard ratio of offspring. So crossing over between homologous chromosomes, it can create new combinations of alleles. So crossing over can separate linked genes, and that's called recombination or recombinant gametes or recombination frequency, right? But it's genetic recombination. Now, the closer together those alleles are on the chromosome, the less likely they are to be separated. And of course, the further apart they are, the more likely they are to be separated. So there's your tetrad, and there's your crossing over, and you get those recombinant gametes with different genetic diversity. So here, the experiment they did with fruit flies, everybody loves fruit fly experiment, and so you have gray body and long wings and black body and vestigial